I've been working with vision for 27 years. I have a medical background. Worked first with brain imaging, then with modeling, and now with simulations. So one of the questions when we are doing computational neuroscience is that can we really say that brains compute? And this is a question of semantics partially, because computing is defined as what person can do with paper and pencil following strict rules. So I guess nobody wants to go that direction, that trying to like conceive that how this would be happen in the brain. But a safer definition I would give that computation is a phenomenon which can be replicated by computer. So if we can replicate a phenomenon in the brain with computer, so we can safely say that there is something computation going on. So why do we need biological realism? So people here know this basically, but our subject is the biology, or my subject is the biological brain. That how does the brain compute? How does it interact with visual environment? But it's also the target of neurology and psychiatry that we want to understand the diseases better. But on the other hand, we can take the engineering perspective that mammalian brain, we know that it's capable of general intelligence. So how does it do it? And that's the other driving force for my work. But then the last question, why computation? That many biological models, including also HBP models, have lots of details, lots of heavy computation, like realizing the simulations, but there is no computation in the target itself. So the cortical column, for example, it's basically doing responses to simple volleys of action potentials. So if I would have, as a clinician, a patient which has, let's say, a square centimeter of cortex left coming to emergency department, so that patient would be basically brain dead, and we would like the next task would be to face the relatives. But if I'm a computational scientist or computational neuroscientist and I have a model of a square centimeter of cortex, I would be like, oh wow, that it's the biggest model of ever. So that's one of the catches here, that cortex is a system, and we cannot just look locally into such a system. This is a timeline of our work, what we have been doing. The dark green means published, and the lighter green means work on progress, and the white means plans for the future. So working with macaques makes a lot of sense, because it is a good model for human vision. They have similar visual behavior. Their psychophysics is similar. They are trichromats. They have similar retinocortical pathways. Their V1 is adapted for foveal vision. They have ventral and dorsal streams, as we do. In monkeys, up to 50% of the cortical surface is primarily sensitive to visual input. In humans, it's like 25. So we are still quite visual animals, and but it's distributed in some 30 distinct areas. So probably in humans it's similar. Although when you go in the extrastriate areas, the division of labor starts to differ between primates and humans. The connectivity matrix is dense. According to this paper, 2014, they showed that about 2/3 of all possible connections are realized in 5 to 6 logarithm units of strength in the presynaptic axons. So it's a lot of mess if you want to model it and understand what it's doing. But anyway, it's a dense graph anyway. A lot of people are studying mice currently, and mice are very accessible with genetic methods, with molecular methods. They have lissencephalic brain, So they have smooth brain. It's easy to measure with many methods, but it's not very visual animal. Its ecology is totally different from primates and humans. And for example, we wanted to check if the Allen Institute data has like biophysics for ventral and dorsal stream, like in monkeys have the ventral and dorsal stream, just to find out that there may be not ventral and dorsal stream in mice, that there are no clear homologies for that. So they have like 9 visual areas in the cortex. And the primate hierarchy is still better understood, and it was studied a lot from 60s to late 90s, and even 2010s there are new papers coming out, but the real, I would say the golden age was up to the late 90s. So of course, maybe for many of us, this is one of the driving forces. Quotation from Richard Feynman, when he died, this was found in his whiteboard, that what I cannot create, I do not understand. So trying to implement the actual computation in a model and trying to limit that model by the biology is what we are basically doing. Of course, acknowledging that the model never is the nature, and it's always, one could say that it's always wrong, but it can be useful. So that's where we are. And I learned the hard way that complex code can hinder development, because I spent a year for nothing to trying to do plasticity and learning in a visual cortex model which I couldn't debug anymore, and then we had to clean up the code, but that's another story. We came up with the CX system originally, and then it became CX system 2 when we upgraded Python 3 and the Brian 2. So it's a high-level simulation tool built on top of Brian 2, and it's primarily meant for cortical simulations. It's driven by 2 CSV files, comma-separated value files, one for anatomics and network structure, and one for the physiological parameters. And then eventually it borrows Brian's engine below it. And you can do different types of simulations. You can do different engines, and of course you benefit from the faster computations only when you have larger models. Basically, it supports oligocompartmental models, so with few pyramidal cell compartments, but this is an open source software, so if I would, for example, need to branch this, I would just interact with the code and make a new branch in the GitHub. So it's available in GitHub. So my student Henri Hokkanen came up with the voucher idea. That's why I'm here, actually. And he suggested that why not take this beautiful Wolfram Gerstner's books, neural models, and implement Jupyter notebooks together with the CX System 2. And he came up with the NeuroDynlib idea, which was the reason, or the voucher which was accepted. And the idea is that we can either analyze existing traces, or we can import models, and then we can export them from NeuroDynlib to CX System, and this way have different types of neural models inside our simulator. Then, so the first thing, if I want to make models of cortical computation, so the first thing, of course, is that I need the tool. That was the CX System 2 to start with, high-level tool to make simulations easy. The second thing is, of course, to have the numbers, the parameters of the primate vision. And I was extremely optimistic when I started this neuroinformatics project with Alessandra Angelucci, who is in Utah, known anatomist and physiologist. And so I thought that, okay, we can do probably macaques and marmosets, maybe cats. And even I had more areas than these three. But then I realized that first, there is no data. For example, for V6 in macaques, not enough data to really talk about quantities of any kind. And then it's huge. The literature is really huge. So we ended up, like, we have more than 350 references here. So basically what I wanted with this paper is to pull together what do we know from the system of these three different areas which would help, as well as it can be extracted from the literature that could be used to limit our models. This is not something anyone with common sense would actually try to do, because it's so complex, all these connectivity graphs and matrices. So this is between areas. Of course, there is certain systematic features here, but it's not plausible to put everything in one model, because, okay, what you get is oscillations if you do that. But then you need to have a very good idea what you are looking at with these oscillations. This is the intra-V1 connectivity graph. Some of the publications have a bit different naming for the same thing, or they made division, let's say, layer 2 and 3 in different ways. So there are like overlapping names, but trying to be as explicit as possible. And the best we could do here is to name them dominant, medium, or sparse connectivity according to best information that we had available. And all this data is available as CSV files, so you get the numbers and the references and connections if you want to use it. So in addition to this layer-to-layer connectivity, there is this system connectivity, so that if you think about the retinocortical pathways, we already have, if we take the magnocellular and parvocellular pathways or their corresponding retinal ganglion cells, we are lucky to have about 90% of the ganglion cells in primates covered. Then we have the blue-yellow sensitive, or mainly blue-yellow sensitive coniocellular pathway. And as you can see, from LGN, lateral geniculate nucleus of thalamus, to V1, already the connectivity is pretty complex. So maybe before the century ends, something like this can be modeled so that it gives more than oscillations. And then when you look how do you go from V1 to V2 and V5, so you have also this subdivision of areas, that you have these modules within areas which are sensitive to different things. And the good thing in macaques is that these are pretty well characterized, that you know a lot of the physiology of these different cells and their receptive fields. One thing which I learned when I was doing this work was that the local horizontal connectivity is very different in these three different areas, that it's much more local in primary visual cortex compared to V2 and V5. So in V5, which is a much smaller area, the horizontal connectivity covers basically quite large part of the visual field. So the substrate for global integration is already much better. And then we did also compile physiological recordings. I think it was like 11 of them, different parameters. And some of them show very different tuning in the three different areas. Like, for example, the spatial frequency peak is much lower, the best sensitivity to spatial frequency in V5 than in V1. So the next thing what we need to make a cortical model of vision is a retina. You cannot put photographs or videos directly to the cortex, even though some people have even done that. But for this, we got receptive field data from Chicilniski lab in the US, and they have done very groundbreaking work on this regard and measuring spike-triggered average data from in several papers. And again, my student was able to find in Stanford servers that, oh, they have data here, and then later asked that whether we can use it, and they couldn't deny anymore. So, but anyway, we are extremely grateful for that data, because then we can, after modeling these receptive fields, we can estimate their statistics, several parameters like the size, the activation of the surround versus the center, and the ellipsoid nature of it, how elongated it is, and so forth. And then, according to these statistics plus literature, we can recreate a retina, which then serves our model to produce the spikes. And we are still examining different methods to recreate, whether it's only statistical, whether we can use generative AI models. But anyway, we end up with spatial temporal receptive fields with this approach. And this is what we need for the modeling the computations in V1 later on. The last work I want to present is what we just recently submitted, and this is the science part. So the earlier was the methodological part. And I want to be a bit provocative. This is a very simple model, but my claim is that you probably all have heard about the predictive coding theory of the cortical operations, that cortex selects a model, internal model, which matches the incoming afferent volley of sensory information. And when this internal model matches the incoming information, then the incoming data is shut down, and then the internal model is what we actually perceive. But why would nature do that? Why not represent the data directly? Why not just make a facsimile of the incoming information? And that might be something that we faced when we're working with very simple models. So this originally comes from Brendel and a company in PLOS Computational Biology 2020. Machens and Sophie Deneve are senior authors in that. So what they showed using this model, basically with 300 excitatory units, 75 inhibitory units, 3 input units, 3 output units. So what they were working on was that how can you teach this kind of a spiking model to replicate the inputs in the outputs, the 3 units, each unit will be replicated only in one of the output units. And what does it mean, these output units? And they claim that you can, the membrane voltage of the output unit would be the error signal in the predictive coding model. And that's very important, because that's a suggestion that what does a biophysical measure, membrane voltage, have to do with computation, which is the predictive coding. So we followed this and we replicated their model, because they shared the code, thanks for them. And we have this spiking neuron model. And after teaching in MATLAB, we get the output, the black curve, to follow the input, the gray curve, with basically leaf units in the system. So our question was that if we do biophysical parameter variation in the system, how does it affect the information transfer? So very simple question. And so we measured coherence between the input and output, both are analog signals, Granger causality, transfer entropy, and as they did in the Brendel paper, the reconstruction error. And all of these could be reflected on the firing rate of the excitatory or inhibitory group. The inhibitory group basically followed the firing rate of the excitatory group, so it's not shown differently. As you can see that when we vary the leak conductance in this case for the inhibitory units and excitatory units on the x-axis, so we get very different firing rates. with the factor of 100 more in the highest firing rates than in the lowest firing rates. And on average, we get more information transferred with the high firing rates. However, transfer entropy and reconstruction error, there are certain methodological limitations in them. The reconstruction error is extremely delay sensitive, and the transfer entropy is very limited because of the high dimensionality of information theoretical analysis. So they start failing in the highest firing frequencies. So the first thing I wanted to do is can we replicate the input, the highest frequencies of the input in the output? And that was very difficult. With the basic model, what we called comrade model, because the excitatory inhibitory units were the same, so I couldn't do it. We went after biophysical values in the different parameters like capacitance and conductance and firing thresholds and so on, and still no, it couldn't follow the highest frequencies. But then, if for the output units, I did two changes which changed the game. So one was that I increased the leak with the factor of 10, which is still barely in the physiological range. And then I decreased the conduction velocity with a factor of 3 to 1 millisecond from 3 milliseconds. So then, in optimal cases, if you really look at the 200 different simulations in parameter space, you can find a sweet spot where the error, reconstruction error, dips to a minimum. But even then, it's typically always at the high firing frequencies. And we know from earlier work that high firing frequencies are the main energy consumption parameter, that action potentials are very expensive energetically. And nature, for sure, needs to diminish or minimize the number of action potentials because our brains is so expensive in metabolic demands, and energy is basically limiting the population size. So it's probably quite strongly limiting the one of the constraints in brain design. So then we wanted to somehow also compare the different metrics, the 4 different metrics, and we came up with this idea of classifying, because the original model teaches the network to put input 0 to output 0 channel, and so on. So if we just take the maximum information transfer, we can do this classification and end up with 10 different replications with different inputs. We end up with a confusion matrix, and we can study the accuracy of the classification of our system. And in this case, for example, when you have all the values on the diagonal, it means that this is perfectly classifying, which means the classifying accuracy is 1. In the worst case, these would be randomly divided, in which case the classification accuracy would be 1/3, because this diagonal is divided by all numbers in the matrix. So we were faced with the surprise. Take transfer entropy, which has methodological limitations, but even with that transfer entropy, we cannot shut down the classification in this 3x3 system. Even when the excitatory firing rates goes to clearly less than 1 Hertz per unit on average, we end up with Bonferroni corrected significant classification in the system. And the same goes for when I pool the data. So it's only when I force the system to be close to not firing at all or totally saturated that the classification accuracy starts dipping. And same for grain chain causality. These two have probably this dip in the middle comes from the methodological limitations in there. But anyway, So then we did a control experiment with 6x6 matrix, 6 inputs, 6 outputs, just to check for the ceiling effect, that this is not a too simple task to deal with, and it shows the same result. So as a conclusion, if transmitted information can be, instead of trying to replicate the input, we make it a model selection. So we have like components of possible, like kernel of possible inputs. And we turn this replication into selection from the existing models. We can save a lot of energy. And this is like the hypothesis I wanted to post here. Then I want to acknowledge the people who have contributed in this, more and more undergraduates recently. But anyway, and then ask response to the eBrain's questions in the final slide. Okay, thank you.